Thanks for inviting me. This is an area I think is really exciting at the moment. There's been a lot of changes over the last few years, a lot of advances in the science. So I wanted to review some of the data on APOL1 mediated kidney disease. Important disclosure, I am the local PI for a research study from Vertex Pharmaceuticals, and I'll talk about that at the end of this presentation. So I'm going to kind of start off with the summary slide or the conclusion slide just to put everything into context. So as we all know, I think there are two risk alleles called G1 and G2 in the APOL1 gene, which are associated with an increased risk of kidney disease in patients of West African ancestry. Now, importantly, there's a range of pathologies that we see with these risk alleles. We see FSGS. We see this pattern of what has been called hypertensive nephrosclerosis or arterionephrosclerosis on biopsy. And there are some specific biopsy findings that might be suggestive of APOL1 mediated kidney disease. We see this commonly in collapsing glomerulopathy, particularly those associated with viral infections. But there's actually a range of other conditions that we see as well associated with these high-risk alleles. Now, most importantly, these high-risk alleles are susceptibility genes. Now, what I mean by that is that the vast majority, or at least the majority of people who carry these alleles will not develop kidney disease. And it's important to remember that. So there are additional factors that are required to develop kidney disease. Then, as I say, excitingly, I think there are some new therapies on the horizon currently in clinical trials. So let's just review apolipoprotein L1 and think about the normal distribution and the function. So APOL1 is a component of HDL. So here's an HDL molecule with sort of the central core of triglycerides and cholesterol ester. Hopefully Ben is excited by this. On the outside, you can see these apolipoproteins. And APOL1 is a minor component of these lipoproteins. Apolipoprotein A1 is actually the major apolipoprotein associated with HDL. But APOL1 is a minor component. And it's produced in the liver and circulates with a serum concentration around 0.3. So the normal functions of HDL, so it plays a role in lipid metabolism, as we know, with reverse cholesterol transport, but also seems to play a role in innate immunity, and in particular, protection against parasitic infection. So when parasites, they take up this HDL, the APOL1 inserts into either cell membranes or lysosome membranes and leads to cell lysis of the parasites. And the one that is particularly notable is trypanosomiasis. So in addition to circulating APOL1, there's a lot of different tissues actually express the APOL1 gene. So this is from the Human Protein Atlas. Here you can see the liver is the highest expression level, as we might expect. Here's kidney beside it, also highly expressed in the kidney. But you can see it's really expressed very widely across a range of tissues. And even within the urinary tract, it's actually interesting, the bladder appears to, at least in this data set, have a higher expression than the kidney itself. So where in the kidney is APOL1 expressed? So this is from the KPMP Kidney Tissue Atlas. And what we can see is that APOL1 is expressed predominantly in endothelial cells and podocytes. And that's what we'll actually be talking about for the majority of this talk. You can see just with this color bar that endothelial cells appear to have a higher expression than the expression in podocytes itself. There's some older data suggested that proximal tuberous cells may also express APOL1, but this appears to be less abundant in that site. So it's very widely expressed, but what is the function of APOL1 within cells? And that's really very unclear. There's some question that it may play a role in autophagy. So autophagy is kind of the regulated clearance of cytoplasmic components. The APOL1 does contain this BH3 domain, which binds to some of the regulators of autophagy. So maybe that's the role. There may be other intracellular roles. But the role is actually uncertain. APOL1 is not widely conserved, so it's only found in humans, gorillas, and baboons. Our closest relative, the chimpanzee, does not carry the APOL1, does not express the APOL1 gene. And there have been humans described with APOL1 null alleles, and they do not develop kidney disease and appear to be healthy. It's unclear if they have an increased risk of infection to trypanosomiasis or other parasites, but overall seem to be healthy. So wanted to spend just a little bit of time on this just to remind ourselves what we're actually talking about when we're talking about these high risk and low risk genotypes. So this is the protein or amino acid sequence of APOL1. We can see it's a 398 amino acid protein. It contains these multiple domains. It has a signal domain, which is required for export out of the cell. It has this pore forming domain, which is important for inserting these pores or channels into cell membranes. And it also has this SRA binding domain. So certain kind of cunning trypanosomes have developed this protein, this protective response called the serum resistance associated protein, which binds to APOL1 and allows them to escape the trypanolytic effect of APOL1. So G0 is considered the wild type or normal allele, but these other two alleles are very common as well. So G1 is two missense mutations, and the net effect is that we actually have two amino acid substitutions. So a serine and a glycine and isoleucine to methionine. So these the mutations are in missense are in linkage disequilibrium. So these two are kind of inherited together on this allele, which is G1. And this is actually the second commonest allele after G0 in the States. The second allele is this one called G2, and this is an in-frame deletion, which results in the deletion of these two amino acids. Again, in this SRA binding domain, and this is the G2 allele. So we have what's called the low-risk genotype. So that's G0, G0, or G0 with one of the high-risk allele. So this is the heterozygous form. And the high-risk genotype is the G1, G1, G2, G2, or G1, G2, essentially having two of the homozygous or having two copies, two alleles of the high-risk allele. And so we use the term renal risk variant for this high-risk genotype or the high-risk genotype. So these risk variants or high-risk genotype have been associated with a range of different kidney diseases. There's a very high association with FSGS and with HIV-associated nephropathy, and you can see these massive odds ratios. There's some association with other forms of non-diabetic kidney disease. There's an association with kidney failure and lupus. There's an association with collapsing nephropathy, collapsing glomerulopathy, and collapsing glomerulopathy and lupus. But important to recognize that this does not carry over into all kidney diseases. So there's no association with IgA nephropathy, and there's no association with the incidence of diabetic kidney disease. So from an incidence point of view, we're looking at a high association with non-diabetic kidney disease, in particular FSGS and some of the viral forms of collapsing glomerulopathy. Now, it's important to recognize, in addition to the incidence, that these renal risk variants are associated with faster progression of kidney disease. So here's three examples of this. So this is an early study looking at FSGS, a small number of patients, 92. But you can see those who carry the high-risk genotype had a faster progression of their kidney disease. This is well-known data from the ASK study. So this was a study looking at blood pressure management and CKD treatment or blood pressure treatment in African Americans. This is a larger study. And again, you can see that whether in subgroups that had either no proteinuria or the proteinuria group, so we can see that in both of these groups, the subjects with the high-risk genotype had a faster progression of kidney disease. And obviously the proteinuria group had faster than the no proteinuria group. And then just sort of an interesting study last year came out in from the CureGN biorepository study. So this is looking at patients with membranous nephropathy, 599 patients. But if you actually look at the number of patients with the high-risk genotype, hard to see here, but we're only looking at 15 patients. But in these 15 patients, there did seem to be a significantly worse rate of progression in these patients. So, so these high-risk variants are associated with an increased incidence of non-diabetic kidney disease, particularly FSGS and forms of collapsing glomerulopathy. Also with some forms of hypertensive nephrosclerosis, and also with an increased rate of progression of several different diseases. Now, I mentioned diabetes, and although the incidence of diabetic kidney disease does not seem to be increased, there is some data that might suggest a faster progression in patients with diabetes. Now, these are patients with diabetes, not necessarily with diabetic kidney disease, but this is data from the CRIC study that Ben mentioned. And what you can see here is that subjects with the high-risk genotype had the fastest decline. And one important thing to note here is that in Black subjects who do not carry the high-risk genotype, who have a lower-risk genotype, still had a significantly faster decline in kidney disease than white subjects in this study. And I think this sort of highlights that this is an important factor in progression of kidney disease in African Americans, but there clearly are a number of other factors involved as well, in particular social determinants of health. So I mentioned that the majority of people who carry this high-risk genotype do not develop kidney disease. So it seems that there's a second hit required. And in many cases, this seems to be an inflammatory stimulus. So the APOL1 gene is highly sensitive to cytokine signaling. And what we see here is when you have this just simple model of an inflammatory state leading to upregulation of APOL1, and here I've put the podocyte, but happens in other cells as well. And this inflammatory state can be caused by a range of different things, can be caused by viral infections such as HIV, or we also saw this with COVID. Lupus is a highly inflammatory state with prominent interferon gamma signaling. Also leads to upregulation or could feed into this pathway. And interestingly, patients with hepatitis C who were previously treated with interferon, a number of these patients who carried the high-risk genotype actually developed collapsing glomerulopathy as well. And this is just a simple in vitro study that was described quite a few years back now, but it just shows you that within 24 hours of exposure to interferon within the podocyte, you have this marked upregulation of APOL1 expression up to 10 times. But I just wanted to contrast this with the endothelial cells, cell line that they used in this study using interferon. It was actually increased up to about 200 times. So really a massive increase in APOL1 exposure with interferon fairly quickly. So what are the cellular mechanisms of injury, particularly within the podocyte? So I think just to summarize this, I would say that APOL1 acts as an ion channel or a cation channel. This field is advancing very quickly, so it's kind of hard to keep up with this. But we have inflammatory cytokines signaling through the JAK-STAT pathways, leading to upregulation of the APOL1 gene, production of more APOL1 protein, and we get insertion of these APOL1 channels into the cell membrane. This leads to sodium influx and cellular swelling and lysis. But we also have different cellular pathways that have been demonstrated. We have effects on the endoplasmic reticulum, effects on mitochondria. This seems to be quite prominent with mitochondrial injury, abnormal mitophagy, release of mitochondrial DNA into the cytoplasm with upregulation of the NLRP3 inflammasome. We get inflammatory cell death. We get problems with autophagy. So a lot of downstream pathways that can lead to cell injury. after upregulation of the APOL1 channel. Now, I focused mostly on the podocyte so far, but just to mention the endothelial cell, in fact, a little bit more than mentioned. This is a great study from Katlin Sustak's lab, and this combined human data with animal models with in vitro and showed, I think, some fantastic results. This comes from the Million Veterans Project, looking at a large number of patients, some 60,000 African Americans, 800 who developed septicemia. And what they found was that those patients who carried the high-risk genotype had an increased incidence of sepsis with an odds ratio of 1.5, but also an increased severity of sepsis. And then they went on to look at a mouse model. So this is just one simple diagram from the paper. So this is an endothelial cell specific expression of the high-risk gene. So as we know, mice do not carry APOL1, so here they have expressed it only in the endothelial cell. And in their model of sepsis with intraperitoneal injections of lipopolysaccharide, what you can see is in this survival curve that after really 24 hours, the mice that expressed the APOL1, there was a high mortality within the first 24 hours. And then they went on to do lots of things in this study, but one of the things they did was cross these mice with a number of other knockout mice with various downstream pathways and demonstrating protection in this same model. They did a lot of in vitro stuff, a lot of omics work. So I recommend this paper for those interested in the cellular mechanisms of injury with these high-risk variants. Okay, so just moving on for the last few minutes to treatment implications. We now have the ability relatively easily, at least at the University of Washington and in many parts of the States, to do genetic testing for these high-risk genotypes. A big question is who we should be testing, and that's a whole talk by itself. So I don't have answers to this, but really just to raise a few questions. Should we be checking only patients who have glomerular disease? Should we be checking patients who have chronic kidney disease with no proteinuria? Should we focus on kind of the diseases with a very strong association like FSGS or collapsing GN? Should we also be looking at patients with diabetic kidney disease? I did show you that there's faster progression. With regard to race and ethnicity, clearly these high-risk variants are found predominantly in people with West African ancestry. So should we try and identify people who are at higher risk for this ancestry? Should we test everybody? With regards to kidney transplant, it seems to be that the high-risk genotype in the donor has a very strong effect on transplant survival. There's some data looking at the recipients, and I know, again, a lot of investigation in this area, but there might be a small effect with recipient high-risk genotype as well. So should we test the donor? Who should we test here? If you do have a high-risk genotype, does that make you ineligible to donate a kidney? And then I think importantly, is there a treatment option? So if you identify somebody with a high-risk genotype, we can certainly enhance our renoprotective therapies. That's kind of the reason that we do a cheaper test. We look for albuminuria. But as I say, with sort of the development or in process development of APOL1 pathway inhibitors, this may become a very important reason to check for the high-risk genotype, and as I mentioned, for kidney donation. Now, one thing I wanted to highlight is that the fact that you carry the high-risk genotype doesn't, I'm going to use a double negative, but does not mean that you will not respond to immunosuppression. And so what I mean by that is there's different forms of FSGS, as we're well aware. Some people have a circulating factor form of FSGS, and even though you carry the high-risk genotype, you can still respond to immunosuppression. So here's an early study from the NIH FSGS genetic study, where they identified patients who were felt to have primary FSGS. They had 217 African-American subjects, and about 56 of these subjects were felt that they may have a condition that would be sensitive to steroids. And so what they found was that whether people carried 2 risk alleles or had what's called the low risk genotype, 0 to 1 risk alleles, the chances of being steroid sensitive were roughly the same. So this is different from patients with what we call familial forms of FSGS, such as Potosin mutations, where typically patients with these sort of Mendelian forms of FSGS do not respond to immunosuppression. You can still respond if you have one of these circulating factor forms of FSGS, but on a background of the high-risk genotype. And then patients who are steroid resistant, again, this is a very difficult group to treat. This was the FSGS clinical trial comparing cyclosporin with mycophenolate. The closed circles are the 2 risk alleles, the open circles are the 1 risk allele. You can tell by these upside down pyramids that people generally did badly. Here's complete remission, partial remission, and no remission. But there were some patients who were steroid resistant but did have a complete remission with cyclosporin, and certainly quite a few who had a partial remission. So we still need to consider immunosuppression, even if we identify the high-risk alleles in FSGS. Now, I think what's very exciting is the possibility for more targeted therapies for APOL1. So again, we have these inflammatory cytokines, which seem to be an important second hit. So maybe we can antagonize these at an early stage. We can inhibit JAK-STAT signaling. I'll talk about a study looking at with baricitinib. There's a molecule that's been developed looking at a small interfering RNA to inhibit the translation of RNA to protein. It's not in clinical trials yet. I'm going to spend a bit of time talking about this molecule, inaxaplin, which inhibits the APOL1 channels on the cell membrane, also on lysosomal membranes and maybe on other parts of the cell. So I mentioned baricitinib. So there's this study called the Justice study out of Duke University. The PI is Ope Olabisi. It's an NIH study funded by Eli Lilly, and it's a randomized, placebo-controlled randomized trial looking at oral baricitinib versus placebo. And this is a molecule that's already on the market and used in rheumatoid arthritis. Looking at proteinuria patients with CKD. So this is already up and running. What I wanted to focus on is the study that I'm involved with. And this is a study looking at a molecule called inaxaplin. So inaxaplin is a small molecule APOL1 channel inhibitor. It's oral. It's given once a day. And again, here you can see these channels inserting into the cell membranes and lysosomal membranes within the cell. And inaxaplin is a direct inhibitor of these channels. The nice thing about this molecule is it seems to be well tolerated. As I mentioned, it's uncertain whether we really need APOL1. And says not many animals do not have APOL1. And the side effects that have been seen so far, mostly headache, nausea, and vomiting. And to my knowledge, there have been no discontinuations of this drug in the small numbers that have been used so far. And it's also an easy drug to use. There is no adjustment for renal dysfunction or renal function needed. And it's not known to interfere with other medications. So this was a pretty exciting study that got published in the New England Journal. And it's not easy to get into the New England Journal with a study that lasts 3 months and a study that only has 13 patients in it with no control group. But this is the data that was presented. So this is 13 patients, mean age 39 years. Their eGFR was 51, and they had 2.1 grams per gram proteinuria on average. And they were treated with inaxaplin, kind of low dose for a couple of weeks, then 45 milligrams daily. And what you can see at 3 months is that in one of these 13 subjects, there was an increase in proteinuria, but in all the others, a significant decrease. And you can see some patients had a 70% decrease in their proteinuria. And if you look at the time scale, so by about 3 weeks, you had about a 25% reduction on average in their proteinuria, which declined further to roughly a 50% reduction in proteinuria overall. Now, obviously, this is very small numbers, but this led to the further study called the Amplitude study. This was actually a phase 2/3 study, so a dose-finding phase moving into the phase 3. But we're now actually in the phase 3 part of the study, which is what we're enrolling at the University of Washington. So this is a multi-center study, many sites across the States, but it's also international. I think in Europe, I think South Africa, Brazil, maybe the Caribbean as well. And so patients who enroll, the initial step is to genotype the patient. So this is for patients who carry the high-risk genotype. So if you have somebody you think is at risk, they'll be genotyped, and if they carry the renal risk variants, they'll then enter the study and be randomized to inaxaplin or placebo. This is on top of kind of standard antiproteinuric therapies with RAS blockade, SGLT2 inhibitors, good blood pressure control. There's a 40-week interim analysis to look at proteinuria, and then say 2-year study, and the final analysis will look at proteinuria and eGFR slope. So just a few points. This is a pharma study sponsored by Vertex. It's a double-blind, randomized placebo-controlled trial. The aim is to enroll 400 patients. And just to emphasize, this is non-diabetic kidney disease. So patients with diabetes are not eligible for this study. The proteinuria range is 0.7 to 10 grams per gram. And we're looking for patients with chronic kidney disease. So an eGFR range between 25 and 75. And as I say, this is a pill that's easy to take. It's a once a day pill, and it's a 2-year duration. So again, just to emphasize, age range 18 to 15, a generous BMI range 18 to 40, 0.7 to less than 10 grams of proteinuria, and an eGFR 25 to 75. And the main exclusion criteria is diabetes. Now, transplant patients are not eligible either. And also FSGS in the setting of a known cause for FSGS. In other words, so people who have background lupus or sickle cell disease, HIV, some other known kidney disease that may lead to FSGS. So this is the Amplitude study. This is our sort of local team. This is my last slide. You can see all the sites down in the sort of east and south of the States. Here we are, our little site up here. We're excited. And the 3 PIs are myself, Bessie, and APOL. And Glenda, who's the Director of External Relations and Patient Engagement at the KRI, is involved with patient engagement. And just to mention Linda Manahan, who is probably the most important person of the team. She's the research coordinator running the study. And then a final thanks to the KRI for supporting this study, and also the administration staff have been very helpful from the Division of Nephrology. So thank you very much, and happy to take any questions.
Thank you, Ashley. Important and evolving topic. Question from Brendan.
That was an awesome talk, Ashley. I had one question. Have you found any sort of credible estimates of how much the risk of high-risk alleles increases the risk of kidney disease? And then number 2, I would just make a comment. There's free genetic testing for APOL1 that's sponsored, I think, by Vertex. So you can avoid the usual hassles with insurance, et cetera.
Yeah, so that's great. Yeah, so there is, as you mentioned, there is free genetic testing, and APOL1 genetic testing is also part of many of the panels that we send off as well. So we're starting to get those results much more commonly. The contribution of sort of the genetic component versus others, I don't have good numbers for that. There's lots of individual numbers looking at sort of patients on dialysis. You know, the sort of frequency of the high-risk genotype is much higher, but I don't have data on the kind of percentage contribution of the genotype as compared to other risks in patients with chronic kidney disease or progression.
Ian.
Fabulous talk on a really exciting area, Ashley. I have 2 questions, if I might. One is, Any ideas why diabetes on its own, without evidence of damage, isn't a sufficient second hit, it seems, to increase incidence of diabetic kidney disease? It's always sort of baffled me. And my second question is, for in the trial, how are you doing the genetic testing and returning the results and things for people who are being screened for the APOL1 high-risk alleles?
Yeah, I am baffled as well. I think, I think diabetes is also an inflammatory condition, but I don't know why you would not get an increased incidence of the disease. But just to emphasize that there is a contribution in progression, that we do see faster progression. Again, that is in patients with diabetes, not necessarily diabetic kidney disease. And so patients with diabetes get other diseases as well. With regard to the genotyping, so they have an initial visit where the genotyping is done. They're consented, the genotyping is done, but before they progress in the study, this is kind of the initial, almost like mini study to test them for the high-risk genotype, and then they're directly informed of that result. And if they pass that initial pre-screening, then they move into a screening visit. So very little happens until they actually have the genetic screening.
And that's just the APOL1 genotype, no other kidney genotyping or anything else, just very specific.
Exactly, just the APOL1.
Last question from Stuart.
Hey, Ashley, great talk. It's always interesting because it started out as a podocyte disorder and now it's a podo and endo disorder. Have you seen anything about, endothelial clinical side effects like TMA in any of these patients? It's just curious because there's so much damage, it seems, to endothelial cells.
Yeah, I haven't, I see a question from Ram, and Ram might be able to answer this better, but I haven't seen an association with endothelial cell injury or TMA in human disease that I can think of, nor have I seen animal models of TMA. So I haven't seen any. I don't know if Ram knows.
Yeah, I guess the most recent question is with COVID, right? I mean, there's some evidence saying that the increased vasculopathy in these high-risk patients may have a link to APOL1, but we don't. I think I agree with you, Stuart. I mean, for the degree of increase of expression of APOL1 in endothelial cells, which is much greater than podocytes, it's surprising that we don't see more evidence of endothelial injury in these patients. And I think that's really kind of intriguing, the relative biology in podocytes versus endothelial cells.
I think for that to study, I think transplant patients would be ideal. The donor with high-risk genotype and being on CNIs and so many reasons to have TMA and faster progression, I guess.
Yeah, that's a very good thought.
Well, ending
As
the
Ashley
discussion.
said, this is an evolving story, and great overview, Ashley. Thank you.
Right, just reiterating, ending this discussion with more questions. I think that's apt. Thanks to 3 excellent presentations and great audience engagement.